So we've identified two major culprits in cancer cells: proto-oncogenes, which suffer gain of function in tumor cells, and tumor suppressor genes, which suffer loss of function in cancer cells. And these two collaborate in ways we don't fully understand as yet to produce the final malignant state. A variety of events can create the genetic malfunctions in cancer, and these all can affect either a proto-oncogene or a tumor suppressor gene. They include gain or loss of an entire chromosome, focal amplification or deletion within a chromosome, as I illustrated for you with the retinoblastoma gene, chromosomal translocations, as I illustrated for you with both the Philadelphia chromosome and the case of the proto-oncogene MYC, a direct mutation in the gene product, which activates the gene product in the case of a proto-oncogene, but inactivates it in the case of a tumor suppressor gene, or defective control of expression, either up or down. Up for a proto-oncogene, down for a tumor suppressor gene. And all of these have been observed in human cancer. Now, how can we authenticate the powerful circumstantial evidence that these genes are involved in cancer? And there are four general approaches. First, what I call guilt by association. If you find the Philadelphia chromosome in every case of chronic myeloid leukemia, which is essentially the case, surely it has something to do with the disease. We can also install replicas of the damaged proto-oncogene or tumor suppressor gene in mice. And this often leads to the production of tumors, and it often leads to production of the same kind of tumor in which the lesion was originally identified in human cancers. And finally, in very recent years, it's been possible to demonstrate that targeting at least a few of these abnormalities with therapeutics gives rise to a therapeutic response, something I will talk about in greater detail in my second and third chapters. Now, how many of these adverse events are required to produce a malignant cell? Epidemiologists were among the first to approach this problem, and by simply charting the incidence of a particular tumor against age, they reached the conclusion that multiple events were required, and by mathematical analysis of the data, they could assign a rough approximation of the number of events required to give rise to certain cancers. For example, childhood leukemia, only a few events were required, which is probably the reason that these diseases occur in children. Lung cancer, 6 or more, and prostate, which we commonly associate with the late middle age and elderly, may involve 20 or more discrete events. If we look at this in a biological sense, we see evolution in miniature. An initial event occurs in a single cell, causing that cell to replicate. And then an event occurs in one member of that population that changes the cell yet again. And this happens multiple times, giving rise finally to a tumorigenic clone. We call this tumor progression. And at the genetic level, the molecular level, we believe, and in fact we have been able to show, that what is happening over the course of time is the accrual of independent genetic events, so that by the end there is a combination of these events that give rise to the malignant cell. Recently, it's become apparent that sometimes a cataclysmic event can occur that shatters an entire chromosome, and the pieces are then stitched back together again in a random order. This single event can create more than one cancer gene along the array of the chromosome. We do not know how often this occurs, but it obviously would be an accelerating event in tumor progression. So we have reached our genetic paradigm for cancer, and it has these components. There are many causes of cancer, but they all work by damaging genes or otherwise disturbing the function of the genes. Both gain and loss of gene function are involved, cooperate in some way in the production of the tumor. Multiple genes are involved, from as few as 1 or 2 to as many as 20 or more, depending on the cancer and the age that it arises. There is a stepwise evolution to malignancy, probably resulting from the selection for favorable properties of the evolving malignant cell. And there is this cooperation among gain and loss of function.